Assalamualaikum. I'm from class AS124 and in this presentation, our group will be presenting about chromatography in forensic science.
My name is Alia binti Ibrahim.
My name is Narkalis binti Khalib.
My name is Fatin Humairah binti Zakaria.
My name is Nur Alia Maisara binti Muhammad Ruslai.
What is chromatography? Chromatography is a technique for separating the component or solute of a mixture on the basis of the relative amounts of each solute distributed between a moving fluid stream called a mobile phase and a contiguous stationary phase. The mobile phase may be either a liquid or a gas, while the stationary phase is either a solid or a liquid.
There are 4 types of chromatography. First, adsorption chromatography. In the process of adsorption chromatography, different compounds are adsorbed on the adsorbent to different degrees based on the adsorptivity of the component. Here also, a mobile phase is made to move over a stationary phase, thus carrying the components with higher adsorptivity to a lower distance than that with lower adsorptivity. The main types of chromatographic techniques that are used in industries are given as the pictures. The second type is thin layer chromatography. In the process of thin layer chromatography, the mixture of substances is separated into its component with the help of a glass plate coated with a very thin layer of adsorbent, such as silica gel and alumina as shown in the figure. The plate used for the process is known as chrome plate. The solution of the mixture to be separated is applied to as a small spot at a distance of 2 cm above one end of the plate. The plate is then placed in a closed jar containing a fluid termed as eluent, which then rises up the plate carrying different components of the mixture to different height. The third type is column chromatography. Column chromatography is the technique used to separate the components of a mixture using a column of suitable adsorbent packed in a glass tube as shown in the figure. The mixture is placed on the top of the column and an appropriate eluent is made to flow down the column slowly. Depending upon the degree of absorption of the components on the wall adsorbent column, the separation of the components takes place. The last type is partition chromatography. In this process, a continuous differential partitioning of components of a mixture into a stationary phase and mobile phase takes place. In this process, chromatography paper is used as a stationary phase which is suspended in a mixture of solvent that acts as a mobile phase. Here, we put a spot at the base of the chromatographic paper with the mixture to be separated, and as the solvent rises up this paper, the components are carried to different degrees depending upon their retention on the paper.
Use of chromatography. The first one is pharmaceutical and clinical testing. Pharmaceuticals companies use chromatography to quantify and analyze compound for contaminants. For example, chiral compounds have 2 different forms due to their atoms differing slightly in pace. One form of chiral compounds is known to be toxic. Vaccination correction is also an application of chromatography. One is law enforcement. Compare sample found at a crime scene and sample from suspect. Test chromatography can be used for a more in-depth forensic procedure. For example, crime scene analysis to test evidence such as blood, hair, and fabric sample to further understand what may have happened at the scene. The next one is environment and chemical industry. Fluoroalkyl substances, also known as PFAS, have become a persistent threat to the human body and the environment. PFAS can be found in the items such as a protective coating on shoes and other fabric, electronics, and even firefighting foams. The next one is hospital. Detect blood or alcohol levels in the patient's bloodstream. Chromatography can be very useful in drug testing and clinical toxicology reports. Chromatography can separate and analyze substances found in urine sample. The next one is molecular biology studies. Hybrid techniques between electrochemistry, EC, and mass psychometry with chromatography are often applied to study of protein, fatties, and nucleic acid. This combination is largely used for metabolomies such as biotransformation, reaction like oxidation reaction, and proteomics such as the purification of plasma protein, hormones, and antibodies. The next one is food and beverage. In the food industry, chromatography is used to separate and analyze additive, vitamins, protein, amino acids, and other nutrition compounds in the food items. Chromatography can also be used to determine expiration dates by distinguishing the number of organic acids present as well as to detect any harmful toxins that may have been added to the food item. In the beverage industry specifically, chromatography can be used to make sure every bottle of a drink prepared is consistent. The next one is security. Gas chromatography can be used to determine volatile gases, furthering safety measures at the locations such as airports and large gatherings with similar safety precautions, like concerts and sporting events to eliminate deadly threats. The last one is petroleum. Gas chromatography is used to analyze finished gas product and refining processes. Chromatography is most notable used in the analysis of natural and refining gas for BTU content and hydrocarbon composition.
Chromatography and forensic science. What is forensic science? Forensic science is the use of scientific method or expertise to investigate crime or examine evidence that might be presented in a court of law. Forensic science comprises a diverse array of disciplines, from fingerprint and DNA analysis to anthropology and wildlife forensics. Chromatography and forensic science. Chromatography is commonly used in forensic science. The chromatography forensic definition adds the law aspect of science and states that chromatography is an analytical process to physically separate mixture with the purpose of identifying compounds from crime evidence during a forensic analysis. There are many different types of chromatography technique used in the forensic chromatography lab, with each having different uses in forensic science. The different type of chromatography used for forensic include gas chromatography, high performance liquid chromatography or we call HPLC, and lastly, planar chromatography. Planar chromatography. Forensic scientists employ two types of planar chromatography, which is paper chromatography and thin layer chromatography. High performance liquid chromatography. Instead of using gas as the mobile phase, this method of chromatography uses a liquid mobile phase. HPLC machine push substance through using a high pressure pump. This machine helps forensic scientists analyze volatile substances such as gunpowder, residue, fibers, and toxins. One of its most common uses is to determine material using explosive. Additionally, many explosive material can wear down during the gas chromatography method. Gas chromatography. This type of chromatography using gas as the mobile phase to move the injected substance toward the stationary phase in order to analyze the mixture by measuring their retention time. This helps forensic lab technicians to identify several factors. For example, one of the most common uses of gas chromatography in forensic is to identifying and determining a pathology conclusion, such as fluid present in a body, analyzing blood samples, or identifying the presence of a poison.
In conclusion, chromatography plays a crucial role in forensic science by enabling the separation and analysis of complex mixture of substances. Its high precision and sensitivity make it an invaluable tool for identifying and quantifying trace amount of compounds in crime scene sample. From drug analysis to detecting explosive residues, chromatography aids forensic investigators in providing accurate and reliable evidence for criminal investigation and also contributing significantly to pursuit of justice.